Okay, let's jump right in. We've got this fascinating material connecting basic chemistry with what's happening inside our cells.
Right. The mission today is really to unpack covalent bonds and, more importantly, to get at the real job oxygen does. It's often misunderstood.
Totally. And you sent over this great point, the big reveal, maybe. Oxygen is vital, yes, but it's not the source of your cellular energy.
Exactly. That energy comes from somewhere else entirely. Oxygen's role is, well, different. Indispensable, but different. More like the cleanup crew.
And underpinning all of this is that core chemistry idea, atomic stability.
Absolutely.
Yeah.
It all comes down to atoms wanting to be stable, like the noble gases. Usually that means getting 8 valence electrons, the octet rule.
Right, the octet rule. And hydrogen's the exception. It just wants 2, the duet rule.
Precisely. And covalent bonds. That's just how atoms make it happen by sharing electrons. It's like electronic glue.
Okay, and the strength of that glue varies. You've got single bonds,
The weakest and longest pair.
then double bonds, like in, the oxygen we breathe. Intermediate strength.
Yep. And then the triple bond, like an aero, nitrogen gas. Super strong, super short.
That strength difference is huge for function, isn't it? Nitrogen and air. With that triple bond, it's incredibly stable.
Extremely stable. It has a massive bond dissociation energy. Basically, it's chemically inert. It doesn't want to react.
Needs lightning or special enzymes just to break it apart.
Exactly. Now contrast that with oxygen, aero. That double bond is, well, comparatively weaker.
And oxygen atoms themselves are very electronegative, right? They really want electrons.
They really want electrons. That makes oro a pretty good oxidant. It's ready to react, looking to grab electrons to stabilize itself.
So that inherent reactivity, or lack thereof, based on the bond type, that's the key.
That's the chemical foundation. Now let's see how biology exploits oxygen's willingness to react.
Okay, so this is where we tackle the energy source myth. The energy isn't from oxygen itself.
No, not directly. The energy is harvested gradually by breaking down fuel molecules like glucose. Think glycolysis, the Krebs cycle, those earlier steps.
Those steps basically strip out high-energy electrons.
That's the payoff.
Yeah.
And those electrons are picked up by carrier molecules.
Yeah.
NADH and FADH are the main ones, like little delivery trucks.
Where do they take them?
To the electron transport chain, the ETC. It's a series of proteins embedded in the inner membrane of the mitochondria. Powerhouse of the cell, right? This is where the magic happens.
Okay, the ETC. So the electrons arrive full of energy.
And they get passed down this chain, like a bucket brigade, from one protein complex to the next. Each step down releases a bit of energy.
It's a controlled fall, energetically speaking.
Precisely. And here's the crucial part. That released energy isn't used directly to make ATP just yet.
Oh, what's it used for then?
It powers proton pumps. It pumps hydrogen ions, protons, from the inside of the mitochondrion to the space between its inner and outer membranes.
Creating a buildup, like pressure.
Exactly. You get this steep electrochemical gradient, a high concentration of protons on one side, desperate to flow back in.
Ah, so that gradient, that potential energy, that's what drives the main energy production.
You got it. That proton flow back across the membrane through a specific enzyme channel called ATP synthase. It's like water rushing through a turbine.
And that turbine action physically makes the ATP.
It slams a phosphate group onto ADP, generating massive amounts of ATP. That's your main energy currency.
It's an amazing system, but it seems like it depends entirely on keeping that electron chain flowing. What happens at the very end?
Good question. The electrons have now passed through the whole chain. They've given up their useful energy. They're low energy now.
They need to go somewhere, right? Otherwise the chain gets blocked.
Absolutely. If they just sat there, the whole process would jam up instantly. No more electron flow, no more proton pumping, no gradient, no ATP.
And this is where oxygen finally comes in.
This is oxygen's starring role. At the very end of the ETC, oxygen being highly electronegative, that electron hog we talked about acts as the final electron acceptor.
It just takes them.
It grabs those 2 spent electrons, combines them with 2 protons from the surroundings, and forms water, H2O.
Wow. Stable, harmless water.
Stable, low-energy water. It effectively removes the electrons, clearing the way for the next batch to flow down the chain. It keeps the entire engine running smoothly.
So without oxygen to clean up those spent electrons, the whole efficient ATP production system collapses.
Immediately, the cell would have to resort to much less efficient anaerobic methods, like fermentation. Oxygen is the key that allows for this massive energy yield through aerobic respiration.
Okay, it's clicking now. Stability drives the bonding type, and oxygen's specific bond and its electron greed make it the perfect final acceptor to keep that biological energy factory churning.
That's the essence of it.
So here's something to think about, building on this. Consider a poison like cyanide. It directly blocks one of the protein complexes within the electron transport chain.
Right, it jams the machinery mid-flow.
How would the shutdown of ATP production look in that case compared to just cutting off the oxygen supply, which only affects the very end? Comparing those two scenarios really highlights why oxygen's specific job as that